the sixth-generation fighter thinks. It pivots on its own spine, slicing through cloud banks and generating forces inside the cockpit that would fold an untrained body. It is the most violent machine ever built for a human being to sit inside. After the mission, the pilot lands. Heart rate is steady. Vision is back. The flight surgeon checks the vitals, finds everything within normal limits, and signs the pilot off for duty. But deep in the nuclei of 60 trillion cells, something is broken that cannot be seen on any instrument panel. In a space so small it would take 1,000 of them to cross the width of a hair, the DNA has been shredded. Every pilot knows the story of G-LOC and grayouts, but the real trauma occurs past the cockpit glass and the cell membrane, down in the nucleus, where the genetic blueprint of the body is coiled and waiting. Inside a single nucleus, the human genome is packed like 2 full meters of the thinnest possible wire. It is wound around protein spools and coiled into dense structures called chromatin, as ordered as a library's card catalog. Under normal gravity, this archive is peaceful. The double helix unzips and rezips, making fewer than 1 permanent mistake for every billion steps it takes to copy life. Sustaining 9 times the weight of gravity for just 4 seconds causes this factory to shake like a building in an earthquake. Centrifugal force pulls on every molecule, and the dense chromatin coils resist that displacement differently than the fluid around them. They begin to slide and strain against their tethering proteins. The double helix stretches. Under this extreme load, the bonds holding the 2 strands together finally give way. The strand snaps, a double strand break, leaving the genetic wire slack and broken. This is chromatin drift. It is a force-driven displacement of genetic material, caused not by chemicals or radiation, but by the physics of inertia acting on biological mass. While the pilot sits in the flight surgeon's office the morning after a sortie, they feel fine. But inside their cells, a repair crew is already mobilizing to handle a crisis. The broken strand immediately releases a distress beacon, a protein called gamma H2AX. It spreads outward from the breakpoint like ink in water, marking the wound so the repair machinery can find it in the dark. This cellular machinery has spent 400 million years evolving to fix strands one at a time. It is now facing a modern environment where hundreds of strands are breaking simultaneously. Usually, enzymes cleanly press the 2 broken ends of a double helix back together, but high-G flight creates dozens of simultaneous breaks, overwhelming the system. Working in the dark, enzymes grab the wrong ends, sealing mismatched chromosomes together and creating a permanent error. This is the panic stitch, an error-prone emergency repair that sews chromosomal translocations into the genome. It is a permanent rewrite of the pilot's blueprint that will be copied into every daughter cell that nucleus ever produces. Over a 15-year career, these panic stitches accumulate. The damage from a single afternoon sortie becomes a lifelong biological record. This chart shows the biological cost. Pilots with more than 500 high-G flight hours show telomere lengths, the protective caps on their chromosomes, consistent with a biological age 8 to 12 years older than their birth certificates. These DNA damage profiles, the specific patterns of breaks and translocations, are statistically indistinguishable from those found in rockers at nuclear facilities. The cockpit has become an environment of mechanical radiation. This is spiral debt. It is a cumulative genomic burden that the body accrues over thousands of flight hours, but lacks the ability to ever fully revise or repay. Engineers are attempting to mitigate this with hydrostatic pressurized flight suits. These are designed to equalize internal fluid pressure, reducing the pressure gradient that drives chromatin drift before it can start. Early trials of these suits show a 30% reduction in distress markers. They slow the accumulation of genomic damage, but the underlying physical trauma remains. This has led to a difficult debate in aerospace medicine. If we can use CRISPR to write genes for more efficient repair enzymes, or chromatin that can withstand centrifugal shear, should we? High-G flight faces a genetic limit. The human body has been paying a biological invoice for speed since the first centrifuge tests in 1935, written in broken strands, panic stitches, and years of spiral debt.